HLA loss is a really important mechanism of post-transplantation leukemia relapse. And here, in order to escape from the strong alloreactivity brought on by donor T cells, leukemic blasts lose through a genomic mechanism, so through the loss of, through a mechanism of copy-neutral loss of heterozygosity, they lose the entire mismatched HLA locus on the chromosome 6. So basically here, what happens is that these blasts become virtually invisible to donor T cells. And this mechanism of post-transplantation relapse is quite important because it accounts for up to 1/3 of post-transplantation relapse after especially haploidentical relapses, and has also been described for other mechanism of post-transplantation relapse, even though with lower incidence. And but the major issue here is that these patients do not have really unavailable treatment. So the first choice usually when dealing with post-transplantation relapses is donor lymphocyte infusions. So the infusion of lymphocyte, delayed lymphocytes. So here, clearly, the infusion of these lymphocytes are totally ineffective because they will not recognize the leukemic blasts. And also immunosuppressant tapering or withdrawal of immunosuppressant drugs is clearly ineffective since they already rely on the graft versus leukemia effect that here is totally abrogated. So there could be the main strategy that we could use is a second allogenic transplantation, but clearly it is not always a feasible option for these patients, especially elderly ones. So basically, HLA loss relapse is a treatment orphan condition. And so here we focused on the use of non-HLA restricted immunotherapies, and in particular in the use of bispecific antibodies, which are, as of today, almost off-the-shelf drug. We wanted to test their possible role in re-engaging T cells against leukemia. We started with seeing basically how was the immune status of patients at transplant. Clearly, with bispecifics, you need basically two things. First of all, patients need to have these T cells and they have to be ready to be engaged again. We analyzed our patients at relapse, and we've seen that the T cell counts were okay, were almost normal in the majority of cases, and also they didn't present an exhausted state, let's say. And second of all, clearly, the second issue with bispecifics, with every targeted immunotherapy, is clear that you need the right antigen to target. So we use the CD33, which is a quite renowned antigen for leukemia. It has also been used in the past with drug conjugated antibodies. And we therefore analyze the CD33 expression on our patients, and we have seen that basically CD33 is really a good antigen to be targeted for an immunotherapy. So with these premises, we started then using this bispecific that in collaboration with Subcleve Lab in Munich. We started by just testing how this antibody performed in a really simple, let's say, outline. So basically with patient samples obtained from leukemia at relapse. So with samples with both donor T cells and the leukemic blasts, we treated these samples with bispecific antibodies. And we've seen how these antibodies were able to induce a rapid and strong activation in T cells. So we then moved into a really more precise HLA loss setting, which was the core of our research, with the use of mixed lymphocyte cultures. We stimulated donor T cells obtained from isolated, sorted from patients, and we stimulated them with leukemia at diagnosis in order to expand an anti-HLA population, T cell population. And then we tested, after various simulations, we tested these T cells against leukemia diagnosis, leukemia at relapse, with or without the use of a bispecific antibody. And what we're seeing is that with addition of a bispecific antibody, well, first of all, we've seen that clearly after several stimulations with a leukemic diagnosis, donor T cells were able to respond to leukemic diagnosis. We already knew that clearly because these were haploidentical donor T cells, so alloactivity was quite strong. We then saw that when tested against HLA loss relapses, in terms of activation and degranulation, there was absolutely no kind of response. So demonstrating that loss of the mismatched HLA is quite an effective post-transplantation, let's say, immune evasion method for leukemic blasts. And then we've seen that by the addition of a bispecific antibody, we had a reconstitution of this activation and degranulation markers on T cells that were almost comparable to when donor T cells were challenged against leukemia diagnosis. So we've really seen unexpected and really striking results in this case. We also tested for a live cell imaging assay the cytotoxic ability of these T cells when stimulated with bispecific antibody, and we've seen say comparable results with a strong cytotoxic effect on leukemic blasts. And this probably is the most important part of our work, we adapted these results in vivo. We tested this bispecific compound with mice. So we generated a patient-derived xenograft of 3 patients, and with leukemia, so clearly with HLA loss relapse. And then after the engraftment, we treated these mice either with donor T cells or donor T cells and the bispecific antibodies. And we demonstrated that while the treatment with donor T cells clearly has no result at all, the addition of bispecific antibodies resulted in really significant reduction of the leukemic burden in these mice. So we demonstrated that being able to bypass the TCR and MHC interaction that is ineffectual in these HLA loss patients can actually unlock T cells and and therefore we're able to fully exploit their cytotoxic activity. So these are quite promising results that clearly might be translated into a clinical setting in the future.